Hello, everyone. This is Cassandra from NOAA Library. Welcome to Library Seminars, the library's online platform to highlight research and ideas relevant to NOAA. Today's presentation is part of our Ocean Exploration Science Seminar series. Today's speaker is Garret O'Donnell, a recently defended master's student from the University of Guam and a 2025 CNAS Fellow with Ocean Exploration. Garrett's research interests are invertebrate systematics, deep sea biology, and population genomics. Today, he will be talking about archipelago-scale genetics of the massive Porites species complex in the Mariana Islands. So let's get this started. The mic is yours, Garret.
Awesome. Thank you so much for the introduction, Cassandra. Thank you for having me today. And good afternoon and half a day, everyone. As Cassandra said, my name is Garret O'Donnell, and today I'll be presenting the work I did for my master's thesis entitled Archipelago-Scale Genetics of the Massive Porites Species Complex in the Mariana Islands. I'm happy to report that I successfully defended this work 5 days ago, and I'm very excited to share the results of this project with you today. Okay, so let's begin with some background and introduction into why I undertook this project in the first place. So we're all probably at least somewhat familiar with the many threats that are occurring on coral reefs with increasing severity and frequency. What may be a little bit less obvious on first blush, however, is that when this stress and mortality occurs on coral reefs, it is not spread equally from coral colony to coral colony. So on one level, there are differences between species. In this example, we can see that the bleaching event that came onto this Acropora thicket left some completely bleached, while the plating agaricids in the foreground and background were relatively unscathed. And while these differences between species might be a little bit intuitive or somewhat obvious, there can also be differences between members of the same species. So in this example, we can see the bleaching event left some within this species of Acropora completely untouched, some in this sort of transitory state of fluorescence, and some entirely bleached. So, a coral's resilience to stress and its ability to survive a stressor is a pretty multifaceted and complex process. As a so called holobiont, coral tissues are subject to several key symbioses that have important implications for their ability to survive a stressor. They have diverse assemblages of both bacteria, archaea, fungi, as well as incredibly important to their survival, their algal symbionts. So, you can imagine that the differences in these assemblages has pretty direct implications for their ability to survive a stressor. In addition, the coral host as an individual can become acclimatized to increasing stress as it goes throughout its life cycle, as well as epigenetic modifications which can occur as a result of stress. And while all these factors are incredibly important for coral survivorship, one of the most important resources at a population and species level remains the genetic adaptation of the coral host itself. So, adaptive capacity is this term that was coined by Gunderson in 2000 as a species' ability to cope with or adjust to environmental change. And how this plays out is that when a population has low genetic diversity, it is less likely to have individuals in that population with the alleles that will eventually become adaptations to a changing environment. Conversely, when a population has higher degrees of genetic diversity, at least some individuals will contain the genetic resources to withstand a stressor at a population level. So this process ultimately boils down to a sort of numbers game on evolutionary timescales. And the point I want to drive home here is that when it comes to the genetic lottery, it pays to have more tickets. So this relationship between adaptive capacity and genetic diversity begs pretty obvious questions about the state of corals on coral reefs. Where do they fall on this sort of spectrum of interspecific diversity, and what do their levels of genetic diversity have in terms of implications for resilience to future stress? So, a big factor that informs the portrait of species genetic diversity is level of population structure and connectivity. Connectivity can be understood through the lens of gene flow. So, gene flow in broadcast spawning corals is mechanized by pelagic larval dispersal, wherein broadcast spawners will release eggs and sperm into the water column that will then fertilize and become larvae and eventually settle back onto either the same reef as the parent or a different reef as instrumented through currents. So, high degrees of genetic connectivity also generally beget high degrees of genetic diversity, as spatially distant populations can exchange genes and increase their localized genetic diversity. Connectivity is also important in reef recovery, as populations can rely on input of larvae coming from external populations to resettle and ultimately restore reefs. On the other side of that coin, we have population structure. And structure is defined by genetic differentiation between populations. This is primarily caused by localized genetic drift that acts differently in a randomized fashion on spatially distant populations. Structure can also aid in localized adaptation because it acts as a barrier for dispersal. So, if spatially distant populations are not interbreeding, they can ultimately adapt to their environments. Levels of population connectivity and structure can vary dramatically species by species, and this was seen in a multi-species analysis by Aaron Hughes in 2004. So what we're looking at is a figure that represents the eastern coast of Australia and 5 different species of coral. And the arrows that you're looking at represent relative magnitudes of gene flow. So red arrows indicate long distance gene flow, blue arrows indicate intermediate gene flow, and green arrows represent gene flow within populations. And what this analysis found is that levels of connectivity and structure vary dramatically from species to species. And this has really important implications for these species' survival because, as I mentioned, it will inform the rates that they can recover from stress. Varying levels of connectivity and structure also have important implications for management because heavily connected populations will react differently than highly disjunct or structured populations to stress. So you can expect that demographic shifts at one end of a spatially distant range will ultimately bear fruit on the other end of a geographic range. Whereas highly disjunct populations may need to have management strategies built around their specific localities. So now that we've gone over some of the more basic biological principles that inform genetic diversity and gene flow, let's talk about what these principles might look like in the Western Pacific, and specifically on the Mariana Islands. Will populations of corals on these reefs exhibit high degrees of population structure, or will the genetic diversity in this region be defined by interconnectivity and gene flow? So we have a couple of hints to answer these questions from the literature. For one thing, the Marianas as a system are highly isolated from other sources of incoming larvae. So Guam, as an example, is around 2,000 kilometers from the Philippines, 1,200 kilometers from Palau, and around 850 kilometers from Yap, which represent the most proximal sources of incoming larvae from external reefs. And additionally, geographic isolation of oceanic islands traditionally reduces genetic diversity. So, because of this isolation, we might expect lower degrees of genetic diversity in the Marianas compared to other regions in the Pacific. Additionally, hydrographic modeling has been conducted in the Mariana Archipelago by the work of Kendall and Pody, and they uncovered that pelagic larvae need to have a pelagic larval duration of upwards of 30 days to reach the Marianas from external sources, which represents a pretty significant physiological barrier to dispersal. Additionally, they highlighted that the general current direction in this region is northward. So there's a northward current bringing larvae from the north to the south. They also pointed to the northern equatorial current, here highlighted this gray squiggly arrow you're seeing in the center of this figure, as a potential barrier to dispersal. So you can see that the southernmost islands of Guam and Rota are in the path of that northern equatorial current. And so Kendall and Pody speculated that that might be a potential barrier to north and south gene flow. Additionally, a single archipelago-scale study of genetic diversity has been conducted in the Marianas, and that was by the work of another student in my lab, Joe Porites. So Joe studied genetic diversity of Porites rus in the Marianas, and his results pointed to limited but still present levels of connectivity between these islands, as well as occasionally very high degrees of clonality in these animals. So, to study these trends and to examine rates of gene flow, we need a study organism. And for my project, I chose to study the massive Porites species complex. Massive Porites are these incredibly abundant and actually occasionally dominant corals in the Marianas. If you've swam, snorkeled, or scuba dove in this area, you have more than likely seen massive Porites. They're distributed across a bunch of different habitats, and they're just about everywhere that you might look. Massive Porites as a term actually includes at least 6 described morphospecies that have been documented in the Mariana Archipelago. However, they're incredibly difficult to distinguish at a morphological level. In fact, the coral species problem, which is a term that describes the genetic and morphological crossing over between species, was actually coined around the massive Porites as a group. So, pretty infamous in their inability to conform to traditional species definitions. There are overlaps in both gross colony and scleri count morphology, which are often seen as the sort of taxonomic silver bullets to distinguish morphological species. Additionally, massive Porites are thought of as being relatively turbid and bleaching tolerant relative to the potentially more fragile species like Eurocropras. They're also known for having high degrees of genetic diversity. So, other studies that have examined rates of genetic diversity have found comparatively high levels. Massive Porites are also thought to distribute primarily through sexual reproduction rather than asexual reproduction. So the figure I'm showing here on the right shows the traditional pathways of coral reproduction. You have sexual reproduction, in which two parents, as I described, spawn into the water column, and asexual reproduction, in which a single colony physically fragments, and then those fragments grow into adult coral colonies. And because of their large bouldering morphology, massive Porites traditionally are assumed to largely reproduce via sexual reproduction. They've also been shown to have variable levels of connectivity, even within the same species on the same locality. And to highlight that, I want to point to the Hawaiian archipelago and Porites lobata. So, in one study, there was a very clear isolation by distance pattern uncovered. And what that means is that you're looking at a figure showing genetic diversity in the y-axis and geographic distance in the x-axis. And as you can see, there's a pretty clear linear positive trend. As you increase in geographic distance, populations become more genetically differentiated. However, a study published about a decade later on those same islands found a more clear isolation by environment pattern. So, in this unrooted phylogenetic tree, the cladal clusters that are highlighted in those colored clouds represent habitat type, and the nodes are colored according to their geographic locality. So, what you can see is that clades are clustering more according to their habitat than by their geography. So, really interesting and different patterns of diversity in this group. Additional work has been done on the massive Porites on Guam. So the first paper I want to highlight is from my academic advisor, Dr. David Kambash, and his work in 2024. Through his phylogenetic understanding of Porites, he found 10 genetic clades of Porites species on Guam. Out of those 10 clades, 6 have, at least in part, a massive growth form. Additionally, phylogenetic species in this region do not conform to morphological species definitions. You can see that through the names of these clades. We have Porites Anae and Evermani-like, and that just reflects that inconsistency between phylogenetic and morphological species. An additional important result of this work was that only 2 regions of the mitochondrial genome were needed to clarify which clade a given massive Porites belongs to. So essentially, you can go out onto the reef, sample a massive Porites, and barcode these 2 regions and determine which genetic clade that massive Porites belongs to. An additional thesis in my lab from Karim Primov used a technique called DD-RAD-seq to study the phylogenetic variation and population structure of massive Porites around Guam. So the results of Karim's work showed virtually no structure on Guam. And this is shown in this PCA that I'm showing here. So the colors of each of those little triangles and circles reflect the geographic locality. And what you would expect if there was a degree of structure is that samples would cluster according to that geography. But as you can see, there's a heavy degree of overlap in both of the clades that he examined. And so as a result, he determined there's basically no structure here. Karim's results also hinted at potentially significant relationship between habitat and clade. So in this phylogeny from Karim's thesis, you can see that the tips of each of the branches on this tree are colored according to the habitat they were sampled in. And there's some degree of segregation. Some clades are only found in some habitat type, and some clades are only found in others. So this brings me to the knowledge gap that I hope to address with my work. Nothing is known about massive Porites species in terms of both their habitat associations or genetic structure at the Archipelago-Scale in this region. We have a couple of hints at the scale of only Guam, but I want to expand those results with my work. So this leads me to a couple of research questions that I hope to clarify my goals with. First of all, what clades do we have and where do we have them? So to answer that question, I'll be using the mitochondrial barcoding methods I mentioned a bit earlier. And also, once we know what clades we have, where do we have them, are there any associations with those clades in terms of their habitat type or the island that they were found on? To answer that question, I employed a nested ANOVA framework as well as pairwise Fisher's exact tests. Additionally, what are the rates of clonality in this group? Are they following the traditional understanding of these large bouldering animals that are not splitting up and producing clones, or are they following some other mode of reproduction? And to answer that question, I'll look at genotypic diversity. Are populations significantly structured from one another? And to answer that question, I'll be calculating the index FST as well as generating a PCA plot. Once we know a little bit more about population structure, is that structure following a pattern by geography? And to answer that question, we'll be looking at isolation by distance. Are there any geographic barriers to dispersal? Is that northern equatorial current that was highlighted in hydrographic modeling acting as a significant barrier? And to answer that, we'll be looking at both structure and pairwise relatedness results. Finally, which populations are harboring the most and least interspecific diversity? To answer that question, I'll calculate rates of heterozygosity as well as generating the statistic FIS. Lastly, I just want to point to this overall trend that I hope you keep in mind as we go, which is how are these trends varying according to genetic clade? Okay, let's get into the materials and methods of how I began to answer these research questions. So first, I want to say that I'm incredibly fortunate to have benefited from a lot of hard work that went into this project from other members of my lab. And a lot of the populations that were included in this study were sampled before I arrived on Guam. So here's an example of all the populations that were sourced from Guam. And the colors of the pins are the habitat types. So we have blue pins representing fore reefs, back reefs represented in red, and river deltas represented in orange. Additionally, two subsequent expeditions to the CNMI provided me with populations from the Northern Islands. So just a couple months before I got to Guam, didn't quite get to go on those, but very fortunate to be working with the material from these islands. So the first expedition gave me populations from Saipan, Tinian, and Rota. And a second expedition gave me populations from Mog, Sarreguin, and Pagan all the way in the north. And here's what that total coverage looks like across all of the islands in the archipelago. As you can see, the habitat types reflect both the ease of access in terms of sampling, as well as the geographic variability of these islands. So the north is defined by pretty consistent fore reef habitat, whereas Guam has a lot more complex habitat regimes. So the next stage is where I come in. And once we have all these samples in the lab, the first step is DNA extractions. So these were performed with the QIAGEN blood tissue kit, and DNA extractions were assessed for their quality and quantity using electrophoresis and a qubit fluorometer. Next, regions of the MT12 and MT20 mitochondrial genome were barcoded with PCR, and you can see an example of successful barcoding with that gel in the middle of the slide. So those bright white bands are successfully amplified barcodes. These successfully amplified barcodes were sent for sequencing at Epoch Life Sciences Center in Missouri City, Texas, and return sequences were used for phylogenetic analysis in Genius. And here are the results of that mitochondrial barcoding. So first, here's an unabridged table of the breakdown of the clade distribution by both island, population, and habitat type. So as you can see, we have a distribution that is according to that phylogeny on the right-hand side of the screen. And there is a bit of polytomy that's occurring between clades 7, 8, and 9. And I address that on a sample-by-sample basis with reference sequences from that Combosh paper that I referenced a bit earlier. So once we normalize relative abundance of these clades by habitat type, some trends begin to emerge. So as you can see, that blue clade, clade 7, is more common in the river delta habitat than it is in the fore reef habitat. But is that trend statistically significant? And as I said, I answered that question with the nested ANOVA framework. So once I tested the significance of these relationships, I did indeed find 3 clades out of the 6 here that had significant relationships with their habitat type, that being clade 3, clade 6, and clade 7. So clade 3 was significantly associated with fore reef habitats, clade 6 was significantly associated with back reef habitats, and clade 7 was significantly associated with river delta habitats. Next, are there differences in cladal assemblage by island? And to answer that question, I used a pairwise Fisher exact test framework. So here's a heat map that shows comparisons of one island to another on a pairwise basis. And the cell color represents the significance in that difference, as well as the asterisk. And the results of this were that 2 islands out of the 7 did indeed have unique cladal assemblages compared to all the other islands in the archipelago. And that is the islands of Guam in the southernmost end of this sampling regime and Mog in the northernmost end. So, some takeaways from this barcoding effort. Certain clades do indeed have a fidelity for a habitat type, but this wasn't found in every single clade that I studied. Additionally, from a geographic standpoint, islands on the extremes do have unique assemblages of massive Porites clades. Lastly, the greatest diversity, as well as an example of a potential regional endemism on a single clade, was observed on Guam. So, now that we know where our clades are located, let's take these results a little bit deeper. As a reminder, here's that cladal distribution, but I want to draw your attention to 2 clades in particular, that being clades 3 and clades 8. As you can see, taken together, they represent the vast majority of the samples that were collected as a result of these sampling efforts. And so, based on the high degrees of numbers in these populations, we can conduct population genetic analysis. So to do that, I sent 2 plates of extracted DNA from these corals to UC Davis Sequencing Center in Irvine, California, which represented 187 samples, as well as 5 technical replicates for a total of 192 samples. UC Davis employed a genotyping through random amplicon sequencing direct, or GRADI, protocol. And GRADI, for those of us who are familiar with population genetic techniques, is a PCR-based genome-wide sequencing protocol that's somewhat similar to RADseq in terms of its applicability and coverage. But instead of using restriction site primers, GRADI employs random primers. And that's in an attempt to reduce the bias that's sometimes associated with RADseq datasets. Okay, let's get into the bioinformatic analysis pathways and finally the population genetic trends. The first step in this process was to ensure that the clades that I inferred through mitochondrial barcoding did indeed represent 2 distinct phylogenetic species. So first, a couple global stats on this data set. 505 million returned reads, which averages to around 2.6 million reads per sample, so an absolute ton of data returned from the sequencing effort. These reads were demultiplexed on a sample-by-sample basis by UC Davis, and demultiplexed reads were returned to the UOG Marine Lab. So the first step was trimming these reads with a program called FastP2, which included tail trimming on both the 5' and 3' end of each read, as well as the removal of poly A tails. Each trimmed read was then aligned to the chromosome-level Porites lutea reference genome via the BWA-MEM2 algorithm, and then aligned reads were filtered according to their mapping quality with the program SAM tools. Filtered and aligned reads were then assembled into loci and eventually SNPs using the STAX, GSTAX, and populations pipeline. So at this stage, samples that had more than 50% of their reference missing were removed from this analysis, and the entire pipeline was rerun. So this sample shearing left us with a total of 147 samples. And this represents a multi-clade dataset with high quality and variant loci only. So around 27,000 loci and around 58,000 SNPs, so about 2 SNPs per locus, with an average read coverage of 73x. So a ton of reads confirming the genotypic identity of these samples. This multi-clade dataset was a Philip file that was then plugged into the program IQ tree to generate the phylogeny that you're looking at on the right. Based on that phylogenetic tree, we can now make a cladal cutoff informed by those mitochondrial barcodes. And indeed, we do have 2 distinct phylogenetic species. So with that result, we can now proceed to population genetic analysis. So the overall goal of population genomic pipeline is to generate clade-specific data sets. So we want to look at genetic variation on a species level, not between species, because that introduces a huge amount of bias in the data set that we're looking at. So based on that phylogenetic inference, we have 71 clade 3 samples and 69 clade 8 samples. These samples were then demultiplexed, trimmed, aligned, and filtered according to that same phylogenetic protocol. And finally assembled into 3 data sets for each clade. So the overall goal of this assembly is to retain as much data as possible while also removing bias from things like the inclusion of invariant sites. However, those invariant sites are incredibly important for calculations of heterozygosity and FIS. So they were retained in this first and largest data set. The next data set removed those invariant sites but kept every single variant site on every single locus. And that data set was used for relatedness analysis. Lastly, and potentially most importantly, a single variant site per locus was chosen for the remainder of this analysis. So that was done to reduce the bias that's brought on from linkage disequilibrium, which occurs when multiple SNPs on a single locus are used that ultimately increases linkage disequilibrium bias. So that data set was used for genotypic diversity, PCA, FST, and structure. Okay, now that we've done a little bit of quality control and data generation, we can return to the research questions that I set out to answer. What are the rates of clonality in massive Porites in these two clades? To answer that question, I generated a cluster dendrogram. So cluster dendrograms are based on identity by state matrices, and they're similar to a phylogeny in what they look like. But importantly, they establish a clonality threshold, above which relationships are deemed to be unique genotypes, and below which relationships are deemed to be clonal genotypes. And this analysis was performed in R using the AgiNet package. And here is what that cluster dendrogram looks like for clade 3. So as I said, pretty clear distinction between the clonal genotypes, which are below that red dotted line, and the unique genotypes, which are above that red dotted line. So some results from studying genotypic diversity in clade 3. Overall, majority of genotypes are unique. So this indicates that sexual reproduction is indeed the most dominant form of reproduction in this clade. However, there are some clonality hotspots, including on the islands of Saipan and Sarigan. On these islands, more than half of the samples that were collected here were originated from clonal distributions. So on Saipan, with the highest degree of clonality, with a unique genotype rate of only 45%. No clonality was observed on Guam, Rota, and Pagan for this clade. Here's what that parallel analysis looks like for clade 8. So a very similar clonality threshold, although we're seeing a bit of difference in terms of the topology of this cluster dendrogram. Overall, though, still majority unique genotypes, which indicates that sexual reproduction is still dominant here. The highest rate of clonality was observed on Rota with a unique genotype rate of 0.81. And coincidentally, no clonality was observed on the same islands as clade 3, that being Guam, Rota, and Pagan. Additionally, clonality is more evenly spread in clade 8 than it is on clade 3. So clade 8's clonality is defined by single pairs of samples that are clonal, as opposed to clade 3, which has a lot of clusters in terms of its clones. So massive Porites do indeed consist of primarily unique genotypes. The difference that we're observing in the clonal topology might suggest differences in the reproduction mode that these clones are derived from by clade. And additionally, geography might be playing a role here. So the examples I've highlighted here show clonality in clade 3 that's derived on 4 reefs. So in this example, I suspect that the clones are probably derived via vegetative fragmentation. So a single colony breaking apart and then each of those fragments eventually regrowing. Even though that's uncommon, I do suspect that that's where these genotypes came from. Clonality in clade 8, on the other hand, is dominated by distribution on the back reef. So the examples here are from the back reefs on Rota. And in those examples, I suspect that polyp bailout might be the mechanism that generated these clonal groups. So high clonality has been attributed to polyp bailout in massive Porites before. And in that example, stress was the primary cause. So these highly stressful, by comparison, back reef habitats might be inducing polyp bailout in this group in these few examples. Turning now to population structure and answering the question of if populations are indeed significantly structured from one another. The first way we can look at that is through PCA results. So PCA plots the most meaningful variation in the data set in an XY schema. So PCs are a certain number of principal components, but the first two, importantly, have the highest degrees of significance, and those are the ones that are plotted here. So the ellipses that you're looking at also represent islands, and they represent 95-degree confidence intervals around that genotypic variation. So a couple of results. Clade 3 does indeed show some significant structure according to geography. Guam, as an example, is incredibly structured from these three populations on the right, which represent the northernmost islands of Sarigan, Pagan, and Mog. Additionally, a northern island clustering pattern is beginning to emerge here, where these northernmost islands are incredibly overlapped, so a very low degree of structure in the north. Here's what the first attempt at generating a PCA for clade 8 looked like. So as you can see, the majority of the variation in this data set is clustered around this central cloud where most of the genotypes are occurring, with the exception of these two sort of rogue genotypes on the far right corner of this graph. So these two genotypes are way outside of the normal range of variance that we would expect from analysis like this. And so for this reason, I determined that they were outliers, so they were excluded for the rest of the analysis. Here's what that rectified PCA looks like for clade 8. Some instant comparisons can be made between PCA results in clade 8 and clade 3. Clade 8 shows significant overlap of every single population to every other population. So there's no two ellipses that are sufficiently segregated from one another. In addition, there are certain genotypes that are outside of their 95% confidence interval ellipses in clade 8. And that just goes to show that there's not a ton of variation in this data set overall. Another way to examine degrees of population structure is through calculations of FST. So FST is this index between 0, which indicates no structure whatsoever, so complete interbreeding between populations, and 1, which indicates no interbreeding whatsoever and complete structuring. In addition to the absolute magnitude of FST, a p-value is generated, which is based on a Bonferroni correction that accounts for the multiple comparison nature of genetic inference. And so here's what those results look like for clade 3. As you can see, the absolute magnitude is a lot closer to 0 than it is to 1, but that p-value that's been corrected with Bonferroni correction indicates that this is significant structure. So overall, low but significant levels of structure between most of these islands. Additionally, there's a lot less structure in the northern three islands than there is for the rest of this data set. So again, that northern group is beginning to pop out. Here's what that heat map looks like for clade 8. So overall, the values of FST are a lot less than they are in clade 3. And additionally, they're often insignificant. So you can see several comparisons here where no significance in the structure was found, especially in the central group between comparisons of Tinian, Rhoda, and Saipan. Now that we know levels of FST, we can answer the question of if these patterns are being driven according to geography. And to do that, we'll examine an isolation by distance framework. So as a reminder, isolation by distance plots physical distance in kilometers against genetic distance, here represented by FST. And the strength of that correlation was tested with a Mantell test. And here are what the results of that analysis look like. And instantly you can begin to see some difference in this trend. Clade 3, pretty simple story. As you increase in geographic distance, you increase in genetic distance. This is shown by both the generalized linear model around this data, as well as the Mantell statistic, which both tell the same story of significant and positive relationships. In clade 8, very much the opposite. So almost no variance in this data set is explained according to geography, only about 1%. And that is indeed reflected in the Mantell statistic as well. So no real correlation between geographic and genetic distance. Okay, turning now to answering the question of if geographic barriers of dispersal are acting in this group. The first way to answer this is by looking at pairwise relatedness. So pairwise relatedness essentially compares one sample to another, and these relationships can be understood in a sort of familial framework. So first degree relatives might reflect a parent and progeny relationship or sibling relationship. Second degree relatives represent cousins. Third degree relatives represent second cousins, and so on. These are continuous values of pairwise relatedness that are binned according to the documentation of the program that I use for this analysis. Important to my results, pairwise relatedness provides direct evidence for population connectivity. So if we see a lot of relatives between islands, we know that these islands are interbreeding. And in clade 3, we see high relatedness, so a lot of familial connections across the entire archipelago, especially in the north. Here again is that northern clustering. You can see that the reefs on Mog in particular are heavily related to one another, as well as between, for example, Mog and Poggen, in which we're seeing high degrees of relatedness between those islands. And here's that parallel analysis for clade 8. So very contrasting in terms of the amount of relatedness. Almost no relatedness. Only 3 genotype comparisons were determined to be even third degree relatives. You'll notice there is a single pair of first degree relatives, but those are those outlier genotypes that we talked about a little bit ago. Another way we can answer the question of if there are any geographic barriers to dispersal is through the calculation of structure. So structure attempts to assess the heritage admixture, or Q, across a theoretical number of clusters, or K. And the best way that I think you can understand these results is through potentially something you might be a little bit more familiar with, like 23andMe results. So when you get those back, you get a certain number percentage of your genetic heritage related to a specific geographic region. And that's very much the process that's going on with these results. So K theoretically could be any number of clusters, but that was optimized according to the Pucomile method in this case. K3 was chosen to be optimal in clade 3. And in this structure plot, each bar represents a single sample. And the color of each bar represents the percent of a given sample's genetic heritage that is applied to a single group. So in clade 3, we can see that there is indeed crossing between islands. Every single genotype in this data set has genetic heritage from one of those 3 clusters. Additionally, there is a north-south gradient that begins to occur. So the majority of the genetic heritage in the north was sourced from this blue cluster. The majority of the genetic heritage in these central islands was associated with this orange cluster. And the majority of genetic heritage in the south on Guam was associated with this dark purple cluster. And here's what those parallel results look like in clade 8. By coincidence, K3 was chosen to be optimal, although that absolutely didn't need to be the case, just a coincidence there. But in contrast to clade 3, there's really no geographic patterns to this distribution. Additionally, there's no real barriers. Gene flow is occurring between all these populations, but there's no real gradient that was as clear as it was in clade 3. So overall, no obvious barriers to dispersal, and that northern equatorial current isn't really impacting distribution here. Zooming out to examine overall patterns of structure between these clades. Clade 3 showed us a pretty straightforward story of significant inter-island structure that follows a strong pattern of isolation by distance. Additionally, the northern island cluster is of note. So the 3 northernmost populations in Clade 3 are clustering together in several independent analysis pathways. This fits with our understanding of what the hydrographic modeling should look like in this region. If currents are flowing northward, you would expect that larvae are retained in the northernmost islands. And this is shown through the high degrees of relatedness as well as low structure. Additionally, Clade 3 has low but significant structure in terms of its FST between Guam and Saipan. And I highlight those islands because we can compare them directly to other species of coral in this region. So Acropora FST between Guam and Saipan was 0.026, and Porites russus was 0.028. And here's what Clade 3's FST looks like. So right in the neighborhood of what we would expect in terms of the magnitude of structure between these islands. In Clade 8, sort of a similar story, somewhat different. So still low and significant levels of FST, but as you can see, the absolute magnitude is about half in terms of the degree of structure that we would expect between these islands. So very different patterns of population connectivity. Additionally, the little degrees of structure that were observed between Guam and Pagan are of somewhat note. So consistently in this analysis, Pagan and Guam were genetically similar to one another, as indicated through their clustering in PCA, as well as their genetic identity in structure results. So theoretically, massive Porites larvae could be making the transit between these 2 islands to maintain that genetic structure. But if that were to be the case, we would expect higher degrees of interconnectivity between adjacent populations than between Guam and Pagan. So some pretty weird currents would have to be occurring to maintain that genetic bridge. This similarity could represent a potential isolation by environment pattern, but that would be pretty surprising, again, given the difference in topology between Guam, which is the southernmost, most inhabited, and geographically, shall we say, interesting, compared to Pagan, which is the northernmost, uninhabited, and somewhat homogeneous. So if it could be an isolation by environment, but that would be pretty surprising. Additionally, Clade 3 showed no pattern of isolation by distance, and that's the first result of its kind in the Mariana Islands. Turning back to the models that I presented at the beginning of this talk, Clade 3 happens to follow more of the first model, whereas Clade 8 happens to follow more of the second model. Turning now to the levels of interspecific diversity between these two clades. To answer that question, I calculated rates of heterozygosity. So observed heterozygosity is pretty straightforward. At any given locus, a organism could be heterozygous or homozygous, and that depends on the amount of reads that are an A or a G, for example, and we can determine if that locus is heterozygous or homozygous. And if we calculate that across the entire genome and then across the entire population, we can derive rates of observed heterozygosity. Expected heterozygosity is a bit more complicated, although it is based on traditionally understood Hardy-Weinberg equilibrium allele frequencies. FIS is then a direct derivative of rates of heterozygosity. So FIS is a population inbreeding coefficient that ranges from -1 to +1, and +1 relates to high degrees of inbreeding depression. Inbreeding depression describes the phenomenon of increased expression of deleterious mutation that's brought on from a ton of homozygosity. So essentially, heterozygous populations are healthier, and homozygous populations might be experiencing inbreeding depression. Here's what those results look like for clade 3. Clade 3 exhibits significant but slight outbreeding as a result of this analysis. So consistently observed heterozygosity was higher than expected heterozygosity in these animals. In clade 8, the exact opposite is true. Interspecific diversity overall is higher, as you can see through the magnitude of those HO and HE values, but the relative to themselves, expected was almost always higher than observed. So that results in a positive FIS value as seen on the right-hand side. Putting those results in context of other Mariana corals, these results are most comparable to other measures of our lab. And you can see some examples of the Acroporas as well as Porites rus in terms of their observed heterozygosity. And here are where my results fall in that context. So closer to the Acroporas in terms of their rates of heterozygosities than Porites rus. Porites rus is a bit of an outlier. It harbors a ton of genetic diversity compared to these other groups. So while it's on average for the Marianas, these values as an absolute are low overall for both of these clades. Additionally, at a geographic scale, the rates of heterozygosity indicate a potential, what I'm calling a comet and tail model of dispersal, where rates of heterozygosity are lowest on the southernmost islands and secondarily elevated to the north. And that's likely because larvae that are spawning in Guam on the southernmost island are being distributed upward through that northern current. Additionally, the contrasting inbreeding patterns between lineages are of note. Outbreeding that's occurring in clade 3 is pretty uncommon in broadcast spawning invertebrates. It's very rarely reported and is thus pretty poorly studied. It has been observed in massive Porites before with a negative FIS value, but that result kind of went unexplained in that paper. Additionally, the high degrees of relatedness that we're observing in clade 3 should not beget outbreeding. If anything, they should indicate that populations are more inbred. So the difference between these two is a bit surprising. Could high degrees of clonality be explaining this result? This has been shown in Acroporas, for example. Higher clonal populations are experiencing outbreeding. Um, and indeed, that does explain some of the variation here. For example, Sarrigan and Saipan are highly clonal and also outbreeding, but that doesn't hold when we look at the archipelago. So Rhoda is experiencing higher degrees of outbreeding despite more unique levels of genotypic diversity. Comparing those results to clade 8, for once, clade 8 is not the problem child. It's the more in line with expectation of the two. So slight degrees of inbreeding are in line with both expectations in the Marianas as well as in broadcast spawners in general. While there is some degrees of inbreeding, these levels are not to the level that would require genetic rescue from external intervention. So turning now to some conclusions including the surprises that were found in this work, as well as the significance, and finally some acknowledgments. The outlier samples in clade 8 that I found. So these were definitely head scratchers. They didn't really fit with what we would conventionally understand is normal in terms of the genetic variance in a clade. For one thing, I want to point out that these samples are actually very well sequenced, only 3 and 5% missingness in their data sets, respectively. So, could they represent a separate species? The PCA result seems to indicate yes, right? They're so far outside of the genetic variance that that usually indicates that they belong to a separate species. However, when we return to phylogenetic inference, they are nested well within clade 8. And this is also returned as a result of cluster dendrogram analysis. So, really, no evidence there for them being separate species. Could they be hybrids? Hybridization has explained a lack of morphological consistency in Porites, which is definitely seen in the Mariana Islands. So, if that were to be the case, these samples should stand out to us in both structure and heterozygosity results. Here's what they look like when they're included in structure results. And as you can see, they take up an entire genetic cluster purely by themselves. And while that does stand out, that actually lends more evidence to the separate species argument. Because if they were hybrids, we would expect around half of their genetic material from clade 8 samples and half from some external species. So structure results don't indicate that these samples are hybrids. Here's what they look like in terms of their heterozygosity compared to other genotypes in this group. Very much in the middle of the pack here. If they were hybrids, we would expect elevated rates of heterozygosity from the external gene flow from a separate species. So overall, these samples could probably be their own project on their own, but these are some of the hypotheses that I tested to explain their variation. Turning finally to the significance of this study. My results provide the first evidence of interpopulation structure between islands in massive Porites. So we knew a little bit about what structure looked like on Guam, and it was uncertain whether or not that would extend to the archipelago. And indeed it does. We see significant structure between these islands. Additionally, the conventional assumptions about adaptability in this group may not apply to the Mariana group. And that's because of the low genetic diversity that I observed in both of these clades. So, if these animals are resilient, especially compared to other species on the reef, that has to be driven by factors besides their genetic diversity. Additionally, the differences in these population genomic trends imply pretty major differences in life history traits between these two animals. Unfortunately, my results don't really give any easy solutions for management. There are some cladal associations with habitat, which might be used as a proxy for species in, for example, restoration strategies or in potential management strategies. But these potentially large intraspecific differences in stress response may occur into the future as a result of these differences in population genetic trends by clade. So finally, I want to turn to some well-deserved acknowledgements. Thank you so much to the advisorship of my thesis committee, my academic advisor, Dr. David Kambash, as well as my internal committee member, Dr. Hector Torrado, and my external committee member, Dr. Pim Bongartz. Everyone at the UOG Marine Lab, all of my lab members, Karim, Darian, Jess, Hector, as well as everybody who did expedition work, including Sarah Lemmer, James Pfeiffer, David Kambash, Joe Perietti, and McKay Reuter. Additional help in the lab from Zane, Rachel, Danny, and Art was incredibly, immensely helpful. Thank you so much to the UOG faculty who have answered my questions over the years and given me guidance, and especially to Guam's very welcoming and supportive community. As me coming into Guam as a budding researcher, I just felt incredibly welcomed by the community that I was able to find there. To my family, friends, and partner, thank you so much for your love and faith in me over the years that I've worked on this project. And lastly, to the staff of NOAA Ocean Exploration, thank you so much for your support in being a Canals Fellow while also working on my thesis. I know it's hasn't been easy, but my mentors, Adrian Copeland and Emily Crum, have been incredibly important and instrumental in making this work happen. So with that, here is the literature that I cited today, and I stand ready to take any questions.
Great. Thank you so much, Garrett. And with that, I have a question to get us started while we wait for people to type their questions in. Um, could you explain why you chose to align your data to the Porites lutea reference genome rather than another species in the genus?
Yeah, that's a great question. So the reference genome essentially acts as a scaffold and a standard of comparison between these two clades. So I chose the Porites lutea reference genome because not only was it the best in terms of its mapping performance and statistical quality after a couple of tests. We tested several reference genomes, including Porites lutea, Porites lobata, and a couple of other species in the genus. And Porites lutea just happened to perform the best. In addition, that chromosome level architecture of this reference genome gives us additional statistical confidence when we're interpreting these results. So basically, it had the highest degree of mapping quality for all the reads in both clades, and that chromosome level architecture was especially helpful in generating some statistics.
Great. Thank you for your response. And then did have an audience question come in. I'm putting that up right now. What other support would you hypothesize for the outliers of being a new species, and what would you name it?
Yeah, that's a great question. Thanks so much, Danielle. Outside of them being a separate species or hybrids, there are not a ton of really clear biological pathways that explain those results. They should conform to the expectations that we're seeing within Clade 8. So it's really unexplained. I would say, yeah, those two, them being hybrids and separate species, are the most likely. And yet, with the test that I performed, we don't really find any evidence. So it's tough because I do think I walked down a couple of analysis pathways to answer that question and kind of came up empty-handed. I'm very open to anybody's theories about what caused that incredible variation that we're seeing. I would really like to drill down and find some answers on that, but it was really outside of the scope of this study to answer that question. In terms of species description, I think, unfortunately, the morphological and genetic crossing overs that we're seeing in massive Porites make it really hard to describe a species based on those two characteristics. But I think Porites marianus would be a pretty good candidate, just describing the incredible diversity of these animals in that part of the world.
Great, thank you for your response. And then we had a question come in the audience chat from Andrew. Hi, Garrett. Curious, what would you propose for next steps in this line of research?
Oh, yeah. So there are a couple of ways that I could still analyze the data that I have. So I mentioned that despite these animals' reputation for being resilient and, relatively tolerant to the stressors that are occurring, um, there isn't really evidence for that in their genetics. And that is true in terms of the reads that were aligned to the Porites lutea reference genome. But another way to analyze that data is by aligning all of my reads to a symbiont reference genome. So there are published genomes that are associated with the coral symbionts, so those microbial assemblages within the coral tissue. And I can align all my reads to that reference genome. It'd be a lot less data because the majority of the DNA in this data set is more than likely from the coral host. But in aligning all of my reads to that symbiont data, I could compare that across populations, across species, to see if there are any important differences that are occurring in symbiont composition with respect to island or habitat type or other variables like that. So I think exploring symbiont assemblages in that way would be really interesting. Again, probably outside of the scope of my master's thesis, but if I had another year or if I was doing a doctoral project, that's probably a place that I would go. But yeah, there's a ton of things that you could still do with genetic data. Another thing that comes to mind is assessing effective population size. So that's a statistic that we can scale according to evolutionary timescales to look at potential influence of glacial extent and retreat. So that might be something cool to do with this data. But yeah, those are the things that come to mind off the top of my head.
Thank you. That's really great. And then our next question, can you use core-like characteristics and morphology for distinguishing most Porites species in the Pacific?
Yeah, that's a great question. So generally, from my understanding of Porites taxonomy, that occurs first at the colony morphology level. So, some diver in the field is able to identify on a colony by colony basis what species Porites, a given Porites belongs to. But within the massive group, that's where the lines tend to blur. So generally speaking, sclerite counts tend to follow that gross colony morphology for a lot of the species outside of that massive morphological definition. So that's largely what I understand for the Porites taxonomic descriptions in the Pacific.
Thank you. And then the next question, in a potential future with changing ocean currents, do you have any ideas on how these clades may be impacted given their differences?
Oh, I love that question. Yeah. So we know that ocean currents are changing as a result of differences in temperature throughout the globe, and as that occurs in regions like the Mariana Archipelago, the northern equatorial current might shift. It might shift in terms of its latitude, north or south. And so I highlighted here that it's really not acting as a barrier to dispersal in the archipelago, but that could change as the northern equatorial current potentially moves north, for example. That could then factor in as a barrier to dispersal. So, those northern islands that are reliant on the genetic diversity of the southernmost islands could potentially see that genetic diversity plummet as a result of not having genetic input from the south. So, there are definitely implications of those changing environments in the genetic diversity of this region.
All right. And then we only have 2 minutes left, and there's two questions to go through, so we might run a little over to answer them both. But the next question is, have you guys looked at or thought about potential microscale morphological differences between the clades? I'm thinking correlate structure or something along those lines.
Yeah, definitely. My project is more from a phylogenetic framework, and purely from a population genetics standpoint. There are some differences between morphological species, especially on a correlate level. But as I said, they don't really follow these genetic definitions. So I'm using the phylogenetic species definition in the work that I used for this project. But morphological species definitions are definitely still very important. So there are some differences, but from what I understand, the work that is done previously, including those two papers that I referenced on Guam, highlighted that the morphology crosses over between these genetic clades. So they're just not agreeing at the end of the day, morphology and the phylogenetics in terms of the species.
All right. And then our final question. You mentioned there were no easy managements according to your results. What would you have need to see in for an easy management solution?
Yeah, that's a great question. Honestly, management is intrinsically complicated, and I definitely don't want to, point to that ever really being the case. I would say, if, for example, the clade distributions were incredibly segregated by habitat type, managers could go out onto a forereef and, very consistently pull in the same phylogenetic species. That'd be incredibly helpful for a restoration framework, right? You know that the forereef on one side of the island would interbreed with the same species on the forereef on the other side of the island. So I guess that would be a more traditionally and simple to understand management strategy. But unfortunately, there's no real simple answers when it comes to management. I definitely recognize that. But yeah, there's just complications in this group, both in their genetics and their morphology. So just layers of complications that begin to build up on one another. So management overall can never be really simple, but in this case, I think it's especially complicated for managers to try to wrap their heads around these species.
Awesome. Well, thank you so much, Garrett, for joining us today and for sharing your work with us. Audience, we hope to see you next month. Thank you for attending and enjoy the rest of your day.